Hello everyone, welcome to our educational webinar titled Data Quality and Standards for Pluripotent Stem Cells. My name is Andrew Gaffney, Senior Product Marketing Manager at STEMCELL Technologies, and I'll be your moderator for this webinar. If you'd like to ask a question during the webinar, please use the Q&A panel or chat box on the right side of your screen, and we will address those questions at the end of the presentation. It is my pleasure to introduce you to our speaker today. Dr. Andreas Kurtz is the founding PI of the Berlin-Brandenburg Center for Regenerative Therapies at the Charité in Berlin and heads its stem cell research and knowledge management lab. His research is aimed at developing stem cell-based tissue models and regenerative therapy approaches, as well as stem cell-focused database development with the goals to provide transparency on cell line quality and accessibility, verify the data, and promote the use of standards. He has coordinated the European Stem Cell Registry since 2010 and is a steering committee member of the International Stem Cell Banking Initiative, or ISCBI. Dr. Kurtz received his diploma in genetics from Martin Luther University in Halle and his PhD in cell biology from the Academy of Sciences in Berlin, Germany. Dr. Kurtz worked as an assistant professor at Georgetown University and Harvard University before becoming director of the German Stem Cell Authority in 2003. And from 2009 to 2016 was a member of the Faculty of Veterinary Medicine at Seoul National University. Let me now hand you over to Andreas to begin the presentation.
Thank you very much, Andrew. And also thank you for giving me the opportunity to talk about this topic of data standards and quality for pluripotent stem cells. And when we talk about data in this area, I would like to emphasize today on three topics. First one is what type of data actually we are talking about when we talk about pluripotent stem cells. The second, what are data standards and why do we need them? And the third topic, how can these data be found, accessed, shared, and reproduced in an ideal world? And we all know that there are many data associated with pluripotent stem cells. They come in different standards and different formats, and many different assays are being used. And today I try to familiarize you with the problems associated with this complexity. So what types of data are associated? If we make pluripotent stem cells, we start usually from a donor, which can be an embryo in the case of embryonic stem cells, or a person in the case of induced pluripotent stem cells or somatic nuclear transfer pluripotent stem cells. And already with this donor are associated data needed to assess the quality of a pluripotent or the characteristics of a pluripotent stem cell line. These are, first of all, the ethics and informed consent information about the donor or from the donor. Can we use this material to make pluripotent stem cells? Can we bank and distribute the cells? Can we derive data from the cells and make them openly or not openly accessible? Then there is important information on the disease, on the phenotypes, the sex, age, ethnicity, and genetic data of the donor. And then we want to know which source cell type did you provide, did you donate or she donate for the derivation of pluripotent cells. The second big area where data are being generated is the area of reprogramming or cultivation and deriving the pluripotent stem cells. Again, the source material is a starting point, but there's the reprogramming method, the clone which is selected, the cultivation, the passaging procedures, the genetic modifications, either through culture, because the cell line is not stable, or through genetic editing means, for example, and the karyotypes. These are all information we derive when we make a pluripotent stem cell line. And there's the final set of data, or not the final, but the third set of data comes from the cell itself, from its characterization. I mentioned here also the microbiology, so the virus load of the cells, not only the reprogramming virus, but also the virus introduced during cultivation or coming from the donor already. What kind of markers are expressed? Are the so-called pluripotent C markers expressed on these cells? What is the genetic background, the genotype of the cell? What is the potency of the cell? Can it differentiate in cells of the 3 germ layers or not. Then the other issues like passage number, if the cells are banked, which batch number is being used, and is there a subclone to the cell derived from a original clone through genetic editing, for example. And the fourth set of data, which is also important, is the data derived from users of the cells. So when somebody uses a cell to differentiate, to build a model for clinical application, then this information is relevant to assess the quality and the robustness of a cell, to reconfirm the differentiation potency, to assess the proliferation, expansion rate, and genetic stability of the cell when it's expanded for example, for clinical application or for disease modeling, and for other applications which now developing quicker and quicker, and the demand for cell numbers for upscaling is very high. So these are the data areas we were talking about when we talk about data for pluripotent stem cells. And I put this table in, which was just recently published. It assesses the usage of human embryonic stem cell lines in the literature. So publications using embryonic stem cell lines, what line is being used, and you can see that 3 lines make up more than 80% of the embryonic stem cell lines being used in all publications. And that number didn't change over the last 10 years. H9, H1, and H7, 80% of all studies embryonic stem cell with embryonic stem cells are done with these lines. Why? First reason is because there's a lot of robust information about performance of these cells available. They're often published. People know what they are doing, what they're having in their hands, what the cells can do or what the cells cannot do. It's robust. It's informed. The second reason is also important. It's a social network reason, and there was a publication a couple of years ago showing that people who know each other hand the cells to each other, and the same cell line is being used by people in these labs, in these networks. So, but I put that on to emphasize the importance of the data in the fourth kind of category, the feedback data from users of the cell lines. Having said that, I would like to come now to the question, what defines the quality of a pluripotent stem cell line? We've talked about donor, about consent and phenotype of the donor. Of course, this is important. Also, the user feedback is important. But what is really important to characterize a pluripotent stem cell line as such? What are the quality criteria? How does it need to be characterized? What kind of data do we need for reprogramming, cultivation, banking assessment? And I focus here on only 5 categories, which are commonly being used. One is the morphology. The cell need to grow in a colony with a certain characteristic. The second is the expression of certain markers, which are typically expressed in pluripotent stem cells. The third one is to show that the cells are actually pluripotent. The fourth one is the genetic constitution. So what is the karyotype? What is the genetic background of the cell? And the fifth one is the authenticity. So is the cell I'm working with actually the cell I think it is? When we go into detail on these categories, we see already a lot of diversity. The first one is the expression of markers. There are, of course, certain markers which are always used, but there are also other marker combinations possible. There are about 30 at least known markers which are typically expressed in pluripotent stem cell in combinations, in different combinations. So the question now is, what is the needed number of markers we need to see? What methods are there certain methods preferred? What marker combination is relevant and minimal? Well, this is the first aspect. We already run into issues in terms of harmonization. The second one is pluripotency. Is it needed to have an actual differentiation of the cell line shown? Is it also needed for subclones of the cell line, genetically modified or genetically edited lines from an already characterized clone? Or is it sufficient to have an indirect assay like pluritest or AP-Pluri-score, which rely on epigenome patterns or on transcriptome assays to suggest that a cell might be pluripotent or not? And there are several other assays developing and being used. The third point here is genetic constitution. So of course it is important to know whether the cell has the phenotype, the genotype we think it has, the carrier, and there are already different granularities we can apply to define genetic constitution. There's whole genome sequencing, there's exome sequencing, arrays, microarrays, SNP analysis, SNP assays, or just karyotype or karyolite or different or similar assays. And the question really is what kind of assay is being relevant for an application or for the basic characterization of a pluripotent stem cell line. And then the last point here is authenticity. There are commonly used assays, 2 of them, which are not interoperable. This is a short tandem repeat SGR assay or a SNP array assay. So both are workable in their own kind of frame, but they're not interoperable. So SGR has advantage that it's a standardized profile. It's already used for many, many years, and it's being validated. The problem is that it's kind of difficult to automatically assess similarity of 2 cell lines. So there's diversity, and there are many data generated. And one point which would help to deal with all these data would be standards. So what are these standards and why do we need them? I already answered the second part a little bit, but predefined standard assays and standards formats have a lot of advantages, and some are listed here. The data generated from standardized assays are highly formalized. Can be strictly applied, and the data standards are not movable. That allows for a very high degree of detail of the data, including raw data, which can be compared and analyzed and stored. There's a comparability, there's an automated validation possible, and automated searching possible among these data standards. Also, validation and certification is robustly defined, and a mass upload of data is possible easily with a universal programming interface, which is relevant for people who have not 1, 2, or 10 lines, but have 500 or thousands of lines with associated data, and these data need to be handled. So these standards allow for interoperability and verification. What is verification? Verification is a term used for data. It means they need to be findable, accessible, interoperable, and reusable. An example for a data standard just mentioned here is a RS format for SNPs. There are ontologies and nomenclatures for diseases, for example, which are also these kind of metadata standards. However, that standardization does not reflect the current situation of harmonization in our field of pluripotent stem cell research. Instead, what we have is the use of all kinds of data types and assay methods. And this is reasonable to a degree because this field is developing, and many new assays. Technology has really revolutionized in the last few years. Many new assays are being developed, and of course, they must have a chance to be applied. So this kind of situation needs to have an inclusive approach, which allows for the current diversity and the current amount of data to be applied. This is, of course, associated with a high effort to enable comparability, for example, by using ontologies which are cross-data compatible. And there need to be reference lines basically for each assay available. And this might be another reason why H9, 1, and H7 are so commonly used, because they can serve as reference lines. These many non-harmonized or non-standardized data systems require manual assessment and development of manual evaluation tools. Validation and certification criteria are variable and must be adaptable. A mass upload of data is requiring individual programming solutions, and there's often a lack of traceability and interoperability of the data. So this situation, the current situation, is not acceptable for the verification goal. And we all know that open access publication, for example, is very much promoted. And this is basically requiring application of verification rules so that the data are findable, accessible, interoperable, and reusable. And this is, in the current situation, very, very difficult to realize. It's also not sustainable in this growing field of application. clinical application in large-scale drug screenings or in disease modeling. At least that is my opinion. Because of that, some people already started to work on the field of standardization and defining quality criteria for pluripotent stem cells. And I'm sure many of you are aware of these. I want just to mention a few examples. And one is shown here, the minimum information about a cellular assay for regenerative medicine. This defines minimum criteria or minimal data sets and quality criteria to assess whether a pluripotent stem cell is applicable in regenerative medicine. It includes data on projects, where the cell will be applied, on the source cell, on the assay being used, on the experimental technology being used for these assays, on the data type, data processing method, also on cell type taxonomy and anatomical location of the source cell, but in our next stage in MAIA-CARM2, also on the cell derived from that pluripotent stem cell line, for which we also don't have unique identifier or even the cell type, let's say, codes. Pluripotent stem cells are being made worldwide and they're being used worldwide, and the cells are exchanged through all kinds of countries, institutions, and labs for many applications. And if these cells don't have a unique identifier, it is very easy to lose information associated with the cell. I have one example here, which is the line H9, which has at least 10 synonyms being used around in the literature. So if you look for data associated with H9, you have sometimes a big problem to find these and to associate these actually with the line H9. In addition, when you put into a system, into a searching system H9, you find all kinds of data which have nothing to do with pluripotent stem cell lines. So there is a risk with this lack of unique identification to lose traceability and the connection between donor and cell and data associated with these cells. There's also a risk, if you don't have authentication methods, of mix-up of cells and contamination of lines. So what we have done is develop a standardized unique identifier or name, which can be generated for each pluripotent stem cell line by registering this line in the Human Pluripotent Stem Cell Registry. It's free, it's openly accessible, and it's an automated tool which can also be downloaded as a program. It contains a number of elements. The first is the institutional acronym of the institute where the cell line is derived. The second is an I, an E, or an S for induced pluripotent, embryonic, or somatic nuclear transfer pluripotent stem cell line. And then is a donor ID, the first donor from which pluripotent stem cell was derived at that particular institution. And these three elements form a unique identifier, which is then annotated by a clone number. So from each donor, of course, many clones can be derived. And a subclone, if a subclone has been derived from that particular clone, a genetically edited, for example, a genetically edited isogenic subclone. So using this, the cell line is, and the associated data is findable and interoperable. For authentication, as I already mentioned, there's no standard. In the pluripotent stem cell field, they are both being used STRs and SNPs, and that doesn't allow interoperability. So even if you have a standard name, a unique identifier, you don't always know whether the donor of two different lines with the same name, with different names, are coming not actually from the same donor, particularly if you buy the source cell from a bank. Many labs can buy this source cell. I want to mention one point, which I mentioned already in the beginning, and this is the ethical provenance and applicability of a cell. The ethics data are highly important to assess whether you can use a pluripotent stem cell line in your research. And the first question is, was there proper informed consent to derive these lines or the source cell? And what was consented to? Was it allowed by the donor to distribute the cell, to bank the cells for an unlimited period of time, to access the genetic data derived from the cells? Or did he say, well, there must be controlled access? What about withdrawal the study or from the donation. And what about incidental findings? When genetic analysis is being done on a, let's say, whole genome sequencing level, and a mutation is found which has a certain risk for disease, does this donor wants to be informed about that? So this is all donor information, and can that be searched and standardized? And we have tried to do that also in this registry to kind of make it searchable, the information provided by the informed consent. And this is just a small snippet of that large ethics form, which is here on data and information. And it says, well, does consent expressly permit collection of genetic information, yes or no? Does consent permit storage of genetic information? Does it prevent the dissemination of genetic information? Or has the donor been informed that the donated biosample or derived cells may be tested for the presence of microbiological agents? So, and then there's a digital answer, and this can then now be screened, compared, and analyzed automatically, basically. And the last point I want to discuss today is findability, access, sharing, and reproducibility of data. To make data is fine, but you want to share them, and you want to make them accessible and reproducible. And I have listed a few points here which are necessary, in my opinion, to make this possible. And the first point here is that K quality and characterization standards for pluripotent stem cells need to be defined. There's no consent currently in the community what these are and how deep these standards need to be applied. For example, what kind of pluripotency assay is sufficient or necessary? What level of genetic scrutiny is needed or necessary? And this probably depends on the application. If the cell is planned to be applied in a clinical study, there's probably a much higher level of genetic scrutiny necessary as if it would only be applied to differentiate a certain cell type and then do some functional studies with it. And also the same, there's no consent in the community on the marker profiles which are sufficient or needed to assess a pluripotent stem cell in its quality. And then the second step would be, after this is consented or defined, is to define the metadata associated with these qualities. For example, what kind of markers specifically need to be assessed, and what kind of assays need to be used. And this is the next point: identify and validate the assays which provide these metadata, which can then also be stored in a standardized way. The next point then is to make these data findable, accessible, and interoperable. And for that, a standard unique identifier is necessary. For example, a standard name or a biosamples ID, which is already an identifier for biological samples. These provide an interoperable name, but we also need an interoperable authentication mark. So STR and SNP analyzers are both fine, but they need to be interoperable. The last point I listed here in this subcategory is to use data standards, vocabularies, and ontologies, which are already for many fields available, and they provide interoperability and kind of work in terms. And also they provide, especially ontologies, provide links to other kind of data areas, which are logically, most of you probably heard of the gene ontology, and there's also ontology for cell types and diseases, for example. Next point, to enable feedback from users. To annotate cell lines is a very important category of data, and also for that, data categories are essential. Otherwise, the feedback will be loaded with a lot of moderation effort. Definitely provide higher robustness and reproducibility of data of lines and research derived with this lines. So the next point is not to underestimate is data protection. And as you may know, genetic data associated with a line can, if they are merged with other data sets, be used to re-identify a person. And that is often not wanted by this person to be re-identified. And these kind of risks need to be mitigated by implementing certain data protection measures. associated with the data. This is most important for genetic data associated with pluripotent stem cells, but also with donor-related disease, health, and other data. And the common approach to that is to make these data not openly accessible, but makes them only accessible after a certain committee has determined whether they are. So I just wanted to mention this data access provisions, which are also depending on the ethics information provided by the donor. Just to put this requirements in perspective, in the human pluripotent stem cell registry, which I already mentioned, there are about 3,000 pluripotent stem cell lines registered. Only 50% or even less have a complete data set registered to this line. So there is a lot of, let's say, incomplete data, and it's not clear based on the consensus in the community whether these cell lines can be certified as being pluripotent if for example, differentiation data are missing, or if certain marker expression data are missing. So it's not a hypothetical problem, it's a real issue. So again, the registry, which provides global visibility of lines, but mainly it provides a way to deposit data associated with a pluripotent stem cell line and makes this findable and accessible. It gets a unique identifier, a biosample identifier as well, and it makes these data transparent. In addition, the registry also provides certificates of validation of data completeness, which are needed by some funding agencies to fund research using these lines. And it also provides, let's say, control or validation for publication of lines for some journals. So I would like to finish with acknowledging the many people who have been involved in defining data standards and promoting the use of quality standards. Glenn Stacey from the UK, Anna Vega from Barcelona, Mark Tognarelli also from the UK, Stephanie, Johannes, and Nancy from my group, Fataru Fujibuchi and Kuni Sakurai from Kyoto, from CIRA, who have been helping or have been actually the driving force behind the MIACARM, the minimal information for assays for regenerative medicine, and Rosario Isazes, who is very helpful in codifying ethics, and many, many others. With that, I would like to close and waiting for your questions. Thank you very much.
Okay, thank you very much for your presentation, Andreas. And that concludes the presentation segment of this webinar. We'll now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. So let's start with our first question. Do you think that there are problems with researchers giving cell lines to each other to do experiments with and donating cell lines between labs? Should cell lines always be purchased from vendors, or is it okay to donate cell lines between labs?
I think it's completely perfect to donate cell lines between labs. The only restriction I see here is that the institution might have a problem with that, and MTA associated with that distribution might prohibit that, and that the donor of the source cell doesn't allow distribution of the line in general. This is mostly provided by commercial or non-commercial vendors or banks. So these lines can then be without any problem being purchased or distributed. Data, sometimes there are issues associated with the data, especially with clinical and genetic data, as I mentioned. So one needs to assess on an individual basis whether the data can also be shared and how these can be shared.
Given that there are a variety of criteria that researchers believe can be used to demonstrate PSC quality, how can a consensus standard be reached, and how can this be implemented?
This is a very good question, and I have no very good answer for that. There are several initiatives, global initiatives, like the International Stem Cell Banking Initiative, or the previous International Stem Cell Initiative, who tried to implement and develop these kind of quality standards for pluripotent stem cells. The take-up in the community was mainly positive. However, there are several constraints to implement this, and one is cost. So it's costly to use, for example, a teratumor assay, and it's also ethically maybe problematic to do that when alternatives are available compared to a pluritest, for example. And if you make one line, that might be okay, but if you make many lines, this cost issue plays a big role. So I see here two things. One is the penetration of these developed consensus, let's say, standards, and its enforcement by, for example, publishers or funding organizations, which must require that these standards are being applied. And the second is a monetary issue, that these, to fulfill these standards, must also be financially feasible. So I know that the International Stem Cell Banking Initiative is now tackling these issues, both of them, and trying to define standards. And but as I said, there needs to be incentive to apply them. And it's not, the incentive is currently not the completeness of the data. It's rather the availability, the ease of availability of the line, and the trust in basically the few data which are available. Does that make that's what I have to say about that. It's really difficult. Maybe one more aspect. If pluripotent stem cell lines become more and more adopted in industrial settings, in the pharma industry, for example, as a basis for drug screening, it's inevitable that quality standards need to be applied. Otherwise, these companies will never use a line if they cannot trust these lines. So I think it will be penetrating once the application is so sensitive that it's just simply a necessity.
Okay, thank you. You said in your presentation that H9s, H1s, and H7s are the most common ES cell lines in the world by publication number. But is every H9 cell line in every lab the same, or are there going to be differences? And if there are, what are the most important differences to look for and check for?
Oh yeah, there are reports that there are, of course, during cultivation mutations occur, and these accumulate in different labs in different ways. So I'm not sure whether the H9 lines in all the labs all over the world are identical. Definitely not on the genetic level. There are H9.2 lines already identified a long time ago, which is genetically different, and I'm sure if you look closely, that will be also the case for other lines. This comes back to the authentication issue of a line, and I mentioned the SGR and the SNP arrays. So it's very important to regularly check that in the lab. There is, you might know these publications that up to 30% of the lines used in a lab are not the lines you think they are. And very often the contamination is with HeLa cells or other very common cell lines. And that is not, for pluripotent stem cell lines, this is no exception. Just to deviating a little bit away from the H9 question.
Thank you. Is there a central resource where researchers can find information on a published cell line's usage? Because negative results are often not published.
Yeah, it would be very nice to have that. No, there's no central resource for that, but I'm very glad that came up, because it's also my opinion that this is very important. In the pluripotent stem cell registry, we thought about implementing that, and this has several problems associated with it. One is who will look at that, and then if a line is registered with certain information and some other person comes up with different information, that there's a conflict, and that needs to be somehow solved and validated. What is the correct information? And somebody has to do that. So this is something we just cannot resource-wise provide. So we don't do that, but I think it's a very important point.
Do you have any suggestions for master cell bank safety testing?
Yes, safety testing this is relatively standardized actually. The safety testing in terms of microbiology, mycoplasma contamination, and viability of the cell is standard, I would think, and there is also no real discrepancy or discussion about. What is a discussion about is the genotyping. So what level of genotyping is necessary specifically for pluripotent stem cells to assess the identity or the quality or genetic stability of a cell line. And here I cannot recommend, my recommendation would be actually to be most stringent and do exome sequencing or WGS whole genome sequencing. It might not be necessary in the moment, but in retrospect to see what the reasons are when a certain assay or application fails, to identify that reason might be very helpful to have these data available. And also to track from the master cell bank to the working cell bank whether there are any issues with cultivation. This might be a very valuable information. But again, this is my personal opinion, and I know there are costs associated with it, and I understand that people don't do that. But since you asked, this is my answer.
Thank you. How often should human pluripotent stem cells be characterized for karyotype and pluripotency?
For karyotype, regularly. And again, here is the issue of granularity. It is known that certain chromosomes, and there was actually a webinar on in this format as well some time ago, that certain chromosomes are prone to have aberrations during cultivation, chromosome 12, 17, certain arms of these chromosomes, for example. And that needs to be assessed and monitored. And I think there's no time limit, but it might be a passage issue. My recommendation would be to passage cell lines to a certain point and then go back to an let's say low passage stock. So I cannot have a definite answer to that. If a cell line is being planned to be used in clinical application, there must be a higher frequency of checking genetic aberrations, of course. And not only for the source cell, which is the pluripotent stem cell line, but also for the clinically applicable differentiated cell. The frequency of that, I cannot really suggest or propose something, but I know there are studies about that by other people who probably can do that, like Peter Andrews, for example, much better than I can.
Okay, and carrying on from that particular question, do you think that there are any genomic aberrations that are acceptable?
That depends on the application. So first of all, I think genomic aberrations that impact the pluripotency are acceptable when the cell can still make the cell line you're interested in. But you need to be aware of that mutation, because it may have other problems causing later on. So for clinical application, of course, there is a line of thought that any mutation in an oncogene or tumor suppressor gene, which has been associated with a certain type of tumor, should not be allowed. And there are, of course, other mutations where we don't know what it causes, even mutations in the reading frame or really impacting the genome or the epigenome of a cell. And here, again, we cannot in the moment say what the meaning of that mutation is, and the risk is, one recommendation would be not to use that line at all because we don't know the risk. The other would be, well, the risk is not so high, and we have put so much work already in that line, we think it can still be used. And I will not take a stand here, but my inclination would be not to use the line when I have a mutation, at least in a causing a gene not to be expressed or expressed in expressing a wrong protein. In non-coding regions, I'm not sure, but I would think depends really on the expression, the effect on the transcriptome, I would think, and on the functionality of the cell. And then there are mutations which are relevant for certain tissues, but not relevant for other tissues. And when the other tissue is the tissue you're interested in, you might just ignore that particular mutation.
Okay, next question. Are there any recommended quality control checks that are different for CRISPR-Cas9 genome-edited cell lines?
Oh, that's such a moving field. CRISPR-Cas9 genome editing, the main issue is off-site effects. And it's very related to the previous question. So first of all, I think one must check for off-site effects when CRISPR-Cas is used for, let's say, sensitive applications. That is, applications perhaps in humans or for certain drug tests, where you compare your CRISPR-Cas or gene editing line with a parental line. And you want to be sure that this mutation is the one which makes the effect and not some off-site integration. So for other applications, it doesn't matter maybe if you have off-site effects or not, as long as you have your targeted gene modified and you can do the functional assay or the disease model you want to do for your particular application. But for clinical application, as I said, I would recommend really a thorough analysis of genome-edited lines in terms of genome and also in terms of its potency. So the genome edited and the offsite perhaps integrations might impact on the pluripotency of the line, and then this line needs to be reassessed whether it's still pluripotent.
Thank you. And should iPS cell lines intended for clinical use be held to a higher level of quality than research use only lines?
Yeah, absolutely. Absolutely. There are several aspects of quality which are not needed for research lines. And these are the assurance of genetic stability and lack of, let's say, mutations with known risk. A much more thorough look at microbiological contamination, and of course, high quality assessment of pluripotency, where it would not be enough for me to just look at the transcriptome to indirectly assume pluripotency. Higher degree of course, authentication, validation, and some other issues controlled by cultivation and expansion conditions. So a more frequent analysis of the genotype, for example.
Okay, thank you. And we have one final question, and that is, if you would do just one characterization assay on a cell line, what do you think is the most important?
For me, it's actually if I say it's a pluripotent stem cell line, I want to know if it's pluripotent, and a pluripotency assay would for me the most important one. if I can choose one. So under pluripotency assay, I mean a biological assay.
Okay, great, thank you. And I think that's all the time that we have for today. I want to thank everyone for attending, and I'd especially like to thank you, Andreas, for your time and for your excellent and informative talk.
Thank you very much.
Thank you everyone again, and goodbye.
Goodbye.